my name is López. I am a group leader at the Spanish National Center for Biotechnology. My lab is a molecular infection biology lab, and we are interested in investigating the molecular processes associated with bacterial infections. We explore cellular compartmentalization in bacterial cells, and more specifically, we investigate the existence of bacterial lipid rafts or membrane microdomains, which contain a specific protein and lipid composition, and we think are involved and play an important role in bacterial infections. Because bacterial lipid rafts are present in almost all bacterial species, now we are interested in exploring the possibility of using bacterial lipid rafts in non-pathogens for biotechnological applications. To explore the industrial potential of bacterial lipid rafts, we coordinate a European project that uses synthetic biology to generate synthetic bacterial lipid rafts, so we can implement biotechnological processes such as antibiotic production of cosmetics, and also detoxification of pollutants. Within this project, we are responsible for engineering and generating synthetic bacterial lipid rafts for biotechnological purposes. Using different tags, we confine proteins in lipid rafts to improve their efficiency. For this purpose, we used Bacillus subtilis and non-pathogenic E. coli, because those are the two most relevant microorganisms that are used in industrial and biotechnological settings. In addition to this project, which aims to generate synthetic lipid rafts, we are also interested in destroying bacterial lipid rafts in bacterial pathogens, because we think that would be an excellent strategy to inhibit bacterial processes that are associated with infections. We are developing new technologies in the lab to better identify and visualize bacterial lipid rafts. To identify lipid rafts, we are studying a lipidomics approach in the lab, which will allow us to identify the constituent lipids. To visualize the lipid rafts, we are now setting up electron microscopy techniques such as cryo-electrotomography or electromography, which will allow to describe and visualize the composition of bacterial lipid rafts at the molecular level. With these techniques, we want to fully understand the organization of bacterial lipid rafts, so we can understand how can we destroy lipid rafts using a battery of anti-Raf compounds that we are now in the process of developing. With these molecules, we want to explore the possibility to inhibit virulent processes in clinical settings.